In this lesson, we're going to look at the sense of vision. Before we take a look at the anatomy of the eye, we need to look at the physical property of waves and how those different properties impact what you see. First of all, the frequency or wavelength. That is the distance from peak to peak. So if you're sitting on a beach and you're watching the waves crash onto the shore, that would be the distance from one wave crashing until the next one comes in. Short waves, or rapid frequency, will give you your bluish colors of the visual spectrum. Long waves with low frequency will give you your reddish colors. So the frequency or wavelength or distance from peak to peak affects the hue or the color that you see. On the other hand, we look at the amplitude, which is the intensity or the height of the wave, how large the wave is. The intensity or amplitude or height of the wave will determine the brightness of the colors that we see. High amplitude, high intensity, very high waves will give you your brighter colors, whereas a low intensity, low amplitude, smaller waves will produce a more dull color. Now looking at the anatomy of the eye. First of all, you have the cornea. The cornea is a clear, protective outer layer. So if you're walking outside and you have a gnat fly into your eye, it will land on the cornea. Another part of the eye, the vitreous humor, or also called vitreous fluid. This is the gel between the lens and the retina. This makes up the largest portion of your eyeball, the white portion. The pupil is that center opening in the eye that's adjustable. It will expand and contract, so either allow light to come in or keep light out. The iris, or the colored portion around the pupil, is actually the muscle that is responsible for the dilation and contraction of the pupil. When you are in a dimly lit area, the pupil will dilate to allow more light to come in. When you're out in bright light, it will constrict to keep light out, which will be controlled by, like I said, the iris. The lens is that transparent structure that sits directly behind the pupil. When light waves come in through the pupil, they're cast onto the lens. The function of the lens is to change shape, which is the process of accommodation. So it will change shape depending on the angle that the image comes in order to cast that image onto the inner surface of the eye, which is the retina. Acuity refers to the sharpness of our vision. When looking at the lens, there are a few conditions that can alter the acuity. First of all, nearsightedness. This is when you can see close objects but have difficulty seeing things that are far away. This is also known as myopia. And if you are a young person and you wear glasses, chances are you suffer from nearsightedness. You need to wear glasses in order to see things that are far away. On the other hand, farsightedness. This is when people can see objects that are far away but have difficulty seeing things that are close up, also known as hyperopia. And as you get older and you enter into your 40s, farsightedness is a very common condition. It's the inability to see things right in front of our faces again, but we can still see things far away. As the light waves pass through the pupil, they're cast onto the lens. Again, the lens will change shape depending on the angle that the light wave comes in the process known as accommodation, in order to cast that image onto the inner surface of the eye, which is called the retina. The retina contains photoreceptors, rods, and cones, which we will talk about here in a moment. This is where transduction takes place. So within the retina, the photoreceptors will transform the light waves into a neural impulse. So transduction is transforming an energy stimulus, here in vision, looking at light waves, into a neural impulse so it can be sent to the brain for translation or interpretation. The center portion of the retina, we call the fovea. This is where your cones are located. The fovea contains the cones, and the rods are in the peripheral part of the eye. So once again, the light waves will pass through the pupil and be cast onto the lens. The lens will change shape in order to cast that image onto the inner surface of the eye, the retina. Within the retina, the photoreceptors, the rods and the cones, will transfer the energy stimulus, the light waves, into a neural impulse in the process known as transduction. The optic nerve will then pick up that neural impulse and carry it up to the brain. As we've learned in previous lessons, all sensory input, except for smell, will then stop at the thalamus. The thalamus will send those impulses where they will need to go to be interpreted. With vision, those impulses will be sent to the occipital lobe. The occipital lobe contains what we call feature detectors. So these special detectors will detect shape, form, color, all at the same time, which is a process known as parallel processing. As I mentioned before, the photoreceptors that you find in the retina are called rods and cones. Cones are located in the fovea, the center portion of the retina, and they allow us to see in bright light. They're active, and also they allow us to see color. Rods, on the other hand, allow us to see in black, whites, and shades of gray. They're active in dimly lit areas, and they are in our peripheral vision. At birth, we are born without cones. Therefore, babies do not develop color vision until somewhere between the first and second month. It is believed that by the 5th month, babies can see the full color spectrum. We do have a blind spot, which we have a gap in our vision. This is where the optic nerve leaves the eye. There are no receptor cells here. There are no photoreceptor cells here. And if you look at the diagram here at the bottom, that shows you where your blind spot is located. Psychologists believe that color is an experience of the mind, the process by which light information is processed through the sensory organs and the brain, and it can be explained by two different theories. The first color theory is called the opponent processing theory. This was a theory proposed in 1878 by Ewald Hering, a German psychologist. Here we have opponent or antagonist pairs of receptors. When one is turned on, the other one is turned off. So blue light waves, when they are turned on, they will turn off the antagonist yellow light waves and vice versa. This theory is useful in explaining why people also perceive yellow as a primary color rather than a combination of other colors, along with the primary colors of red, blue, and green. Supporters of the opponent processing theory use the after image to support their theory. If you stare for a few seconds at the center of this slide, at the yellow plus sign, when I go to the next slide, you will see the antagonist pair, and you will see blue in place of where the yellow is right now. Here is another example of the after image. Stare at the X for a few seconds, and then when it goes to the next slide, you will see a green X in its place as the after image. The second color theory is called the trichromatic theory, tri meaning three. According to this theory, we have three main types of cones, red, blue, and green. And all of the colors, millions of color variations that we see, are made up of combinations of these three different types of cones. This was first identified in 1802 by Thomas Young. Later on, it was revised by Hermann von Helmholtz in 1852. So sometimes the trichromatic theory is called the Young-Helmholtz theory. When we say someone is colorblind, they are usually deficient in either red or green cones. And it's not that they can't see color, they just see different color variations. For example, someone who is deficient in red cones may see shades of red as shades of brown. Someone who is born with no cones and is completely colorblind, that would be referred to as monochromacy, and that is very rare. If you click on the hyperlink color deficient vision, that will take you to the Ishihara test, which is a test to determine which cone someone would be deficient in.